Okay, good morning, ladies and gentlemen. Let me start directly the presentation of our co-facility. Here's the content of the talk. Basically, it's probably the same as the previous speakers used for content of their talk. And let me start as the introduction. Okay, don't worry, that was just an introduction, so you could briefly see what we are working on. So we are mostly using Afm microscopes to study biomolecules, nanomaterials, but also living cells, cell clusters, and with the scale up, also tissue sections, for example. But not only the bio-AFM, we are combining the AFM, for example, with the optical microscopy, fluorescence microscopy, confocal, but also with the Raman microscopy or multi-electrode array to study the cell physiology. Or we are running some biosensor platforms, some AFM extensions, like, for example, fluid FM and nano indenters. So we will now talk about it in more in detail. So regarding the equipment of the co-facility, traditionally we are running the large AFM systems from JPK, which is JPK NanoVisat 3 and 4 version now installed this year. The new AFM was extended with the fluid FM, technology, which is like microchannel microfluidics technology inside the AFM cantilever, which can either aspire or aspirate picoliter volumes, or you can use for large processing of mechanical properties, for example, the nano indent of Biosoft from Brugge. For the largest resolution, for the high resolution, we used AFM microscope from Brugge, dimension FastScan, or multimode 8 version HR, or Integravita from NTNBT. For chemical mapping or chemical characterization of the samples, we are using Renishaw Raman microscope combined with the AFM. And we were talking about the SPR biosensor platform, or now we are running also the quantum dot characterization reader. And because we are running many experiments on electrically active cells like cardiac cells or neuronal cells, So to offer the possibility to read the field potential of these cells, we are offering multi-electrode or micro-electrode array from multi-channel systems. And traditionally, from the time of Peter's Plado, we are running some electrochemical for bioelectrochemistry instruments. And here is a list of the service based on the instrument we have. We are offering this basic list of the services for the users, basically imaging and mechanical properties, starting from really single biomolecules up to the tissue slices. Then you can extend this service list with the Raman microscopy for chemical characterization, electrochemical measurements, or electrophysiological measurements, SPR biosensor, and scanning of upconverting luminescence. Always we can discuss what is like useful for you or which could be useful, and we can like find out the combination of methods. So now for more, I would like to talk about the applications or real examples of applications. So starting from the left, this is traditionally imaging of single molecules, or here you can see some nanorods. So we are also running the characterization of nanomaterials or combination of nanomaterials with biomolecules. Here you can see the maximal resolution of the micro AFM technology, which is basically the double helix of DNA. It's like really, I think, cutting edge. The advantage of the AFM imaging is that this imaging is running in so-called semi-physiological conditions. So no drying, no staining, whatever. You just prepare your sample on a flat surface. You do it for example, in a phosphate buffer, in buffered conditions. You can heat it up or cool down. You can in situ add some molecules or remove or change the conditions, and all this imaging was done really in buffered conditions, which is one of the main advantages of the AFM comparing to the other microscopies. Then, as I usually say, the AFM is not, usually people think that the AFM is only the imaging, but it's not true. So many, or maybe even most of users, they are coming and they are asking for so-called nano-indentation. Nano-indentation is simply a stiffness mapping of, for example, living cells, which is very much appreciated by the users. And then the top class, very affinity interactions mapping, for example, which is very time demanding and user demanding. So because the time is limited, let me go just briefly through the examples. I like this project. It's quite like old, but it was the starting point for me here. If we are talking about the combination of the biomolecular imaging, so you can see how the complex molecular complex is like originating on a surface, but that's not everything. It's just the structural information. But more important for the user, it was Lukáš Kubala group, was how the stiffness of the molecules or the complex, starting from the hyaluronate, which is very soft, creating the complex with the myeloperoxidase enzyme, it's stiffening 20 times. And it's very important physiological information. Maybe I will skip this. It's just like introduction, like that for the users, not the structural information coming from the AFM is not usually that important, but what they are looking for is the stiffness mapping, how the stiffness is changing during the time, for example, in this game, how the stiffness is changing between the wild type and knockout cell line and so on. So then you can make some video how the cell is changing during the time. There is always some story behind it. We don't have time today, I guess, to talk about it. And also the great advantage of this large AFM systems that you can combine in situ, really like overlay, that you make overlay images with the, for example, with the fluorescence microscopy. So you can, in one slot, get the elasticity map, the height profile map, which could also give you some information. And then here the cytoskeleton was stained in situ, so you can follow the changes in a cytoskeleton structure. And so you are getting like free information in one experiment, and you do it in timeline, how the cell is developing, for example. New from this year, it's extension of the AFM technology with so-called fluid FM, which is very fancy. I like it. Honestly, we are just starting to use it. It's like microfluidics inside the AFM cantilever, where you can use it for many applications. It's shown here. For example, like you can spot, for example, some picoliter volumes. You can aspirate a volume from a single cell and so on. Or you can study the cell adhesion on a surface, which is also very useful for many users. As I said already, I guess we are combining now the AFM with the multi-electrode array, where you can measure. Here it's shown that you measure the blue, it's like the field potential, and the orange is the mechanical data coming from the cardiac cells. So if you combine this, which we can do, this gives you information about the mechano-electrical coupling and decoupling, which is connected with many heart diseases. And I will try. Sorry, no. There's a video, but okay. Before we got this fluid FM technology, we were using to study the cell adhesivity on different type of surfaces. We were using so-called scratching from the surface, using this special cantilever, for example. And traditionally, we are running with the Vladimir Retrekl group this biomechanics on cardiac cell clusters, stem cells, patient-derived stem cells related to heart diseases. And if you connect the cell clusters with the AFM, and I cannot run the video simply, sorry, but then you record the biomechanics in real time. And coming with the sample size upscaling, let's say, here are the examples that you can run, for example, the AFM nano indentation even on a plant cells, for example. So here with Eva Benkova lab, some hypocotyl characterization, biomechanical properties, again comparing wild type and some, knockout, for example, somehow changed lines of the plants. Very nice project. Now running downstairs in the lab still. Again, like correlative microscopy. Here is the polarized microscopy, and you can see the collagen fibers inside the liver samples. And then you can characterize them, not very well visible, unfortunately, on the screen, but you can see here the mechanics of this collagen on all the liver samples connected with the fibrotic diseases inside the liver samples, for example. Continuing with the new technology, Raman microscopy combined with the AFM could give you, for example, information about the nanoparticles, microparticles. If they are okay, you can get the structure of the particles even on optical microscopy, electron microscopy, or AFM microscopy. Still, if these particles should be loaded with something, some compound, you are not simply getting this information from any microscopy. So if you include the Raman microscopy, then you can see that really these nanoparticles are really loaded with this. So this can be one of the applications, for example. Another application we are running now, here is the bone characterization. The AFM gives you the topographical and mechanical information about the bone sections, but the Raman microscopy gives you nicely resolution about the calcification state of the bone. So all together gives you a complex overview of the bone composition connected, for example, to arthritic states. Similarly, can be applied on Alzheimer's disease fibers, for example. Then also the Raman gives you some chemical information. I think the budget is not interesting for today. People joining us, and yeah, we are trying. I'm in my position. I'm from 2000. 19. So I'm trying to increase the number of the users significantly. I'm also trying to attract people to sign for the CISB project, as was mentioned. Probably if you sign for the CISB project, you fill up the application, we consider the application, and if it's approved, then you are getting this lowered price to 10% of the payment. So we attracted, during the last 2 years, we attracted 26 projects, which some of them due to the corona probably even haven't started yet, because people are still preparing samples, so I don't know, starting. Yeah, for the last 2 years, so we are not maybe that productive regarding the impacted publications, but for the 2019-2020, the acknowledgement of the code facility was found in 33 papers, which some of them are listed here. So we have 2 minutes. We are also organizing, because the number of staff in the code facility couldn't really fulfill all the requirements from the users. So the practice should be that we are teaching the users of the code facility to use the instruments themselves or partially themselves, or even, for example, process the data, because the processing of the data is at least half of the work we are doing. So we organized this five workshops with how many? Over 200 participants together. One of them was cancelled, was just in the beginning of the corona. Yeah, we were also joining the user survey. We found most of the people are satisfied, but there are still some who are not satisfied, and we know who is it. And it doesn't mean we will punish them, but no, it's vice versa, that we finished the project as they are now satisfied. Okay, so this was invitation for some of the workshops. You can see that usually the one-day workshop we are organizing, it's like in the morning we go for the lectures, then we go for lunch, and after lunch we go, to keep everything running, we go for hands-on sessions, which is quite like complicated because the space we have, it's quite like small, but we can split the groups and change people inside the labs. We can do it anyway. We are opening also like offer for the industry. So we selected 4 main topics which could be maybe interesting for the industrial partners. But for example, honestly, I need to say, for example, the drug testing on cardiac cells, we need to somehow like cooperate with the research group of Vladimir Otekov, for example, because we are offering technology, but we need someone to offer the cardiac cells, but we can always find some compromise, I guess. We are consistently improving the booking system for the users. And regarding the data sharing, I very much prefer this medium storage of Masaryk University, which is nicely available for the Masaryk University employees, students, or whatever. For the external users, it's still quite problematic. But for these people, I'm using the OneDrive, which is also good, but not that perfect like this medium storage. I think we are trying to get implemented in the correlative microscopy project, for example, because most of the applications we are running are like really correlative, like we are always combining at least 2 kind of microscopies. And regarding like the education, teaching, development of the users, that I see very promising to open the educational project of microscopy for the students and young researchers, where I feel like that the people, they need more education really from the basics of the methods we are running. That would be very, very promising. So, okay, acknowledgements mainly. Let me mention Peter Sklado here. who found the idea to start the core facility of nanobiotechnology. Yeah, that was left one of the users, and I would wish that everyone would be satisfied like him. When he was leaving to Prague, just left the notice, thank you very much for everything on close to the microscope. And I would like to ask all the users of the core facility or the facilities of the SATEC to mention the acknowledgement to the project, which is supporting us with the great instrumentation. I can say really thanks to this project, CISB, we are equipped with really top instrumentation I would never dream about even like use before. So thank you for your attention.
Thank you, Honza. And there's space for your questions.
I have a question about the live cell imaging. So how fast can you image cells, and what resolution does it damage them over time?
Well, it depends which kind of cells you are running. So mostly, if you have something like
Fibroblast cells.
Fibroblasts are fine. I mean, if we are talking just about the imaging, it takes, I don't know, like 10 minutes. If we are talking about this nano indentation, it takes like longer time because you need to in each point. So then it depends how many points you need to get from the cell. So then you can run from 5 minutes to 40 minutes. So, but still, you keep the cells in a petri dish heated. You even have the CO2 chamber, and you work on them. And we use the spherical probe to be maximally gentle and very low forces. So usually, cells like fibroblasts, they survive days even this imaging. Of course, they are some knockout cells from Giancarlo, for example. Then we need to be gentle. Always different, but
A question about the Raman. Is it possible to focus the Raman to some part of your sample somehow?
Yeah. I mean, like, yeah, because it's really like a upright microscope with selection of the objectives. So you can select from 5x to 100x, and then you just run, or you select an area, and then you go, and then you collect like Raman spectra, like point by point.
Okay, so you can do
scanning
point by point.
And then there is either like that you scan like long range spectra, the whole spectra, and then it like takes long time, or you select just small area of the spectra, and then the monochromator can keep the position, and then you are running quite fast. So it's called, I think, HR mapping, and then you can still see the lines how they are like rising. And it's quite fast here.